this computer. All right, so it looks like it's starting to record. So where was I going with that now? Oh yeah, so what our plan is for today. So chapter 13 is it's less intensive in terms of like naming or certain types of reactions. More of this is like an introduction to this type of molecule. Carbohydrates is a kind of like a big family, even though we mostly think of like table sugar or starch. And we'll focus a lot on that towards the end, but a lot of this is just going to be basic theory. And as we go through, and it's going to make out little, more like an emphasis points, like these are the things that, you might see in an exam or something like that. So that will be our plan. So why don't we get started? So even just defining what carbohydrates are, right? If you just break down the word, we can already see there's carbon and hydrogen. That's going to be basically the take-home message. But it's not that necessarily all things made out of carbon and hydrogens are carbohydrates. Specifically, they meant that they can be used as food for organisms. And so they're a major source of energy in our diet because we know fat and protein will be the other two, right? And even though it's not really in the name, we see our carbon, our hydrogen, but it also contains oxygen, right? And what we call these when they're made, usually more sciencey word, is saccharides, right? So you might have heard something like saccharine sweet is like a phrase that people use to say something that's overly sweet. It's come from the word saccharide, which I believe is Greek, which translates to sugar. So even though we think of sugar as just like table sugar, scientists use it more of like a broad term. So we'll talk a little bit more later too, like, when you have DNA, sometimes you talk about the sugar backbone, right? So we need to expand what our definition of a sugar is. So basically that's what we were just going over there, but where does it come from in nature and how do we eat that? Well, carbohydrates need to constantly be regenerated because we're using them as fuels and organisms use them as fuel. And where does that come from? This goes all the way back to like grade school science, but photosynthesis, we have our plants that will take carbon dioxide and water, and photosynthesis will then make glucose and then produce the oxygen that we breathe. And then so this would be more like plants go in the forward direction. And then respiration would be something like humans. Though respiration, even though we think of it as breathing, is technically the process of electrons that are flowing that generate the energy. So even though we call it respiration, maybe it is the medical term to breathe, but in science, especially in biochemistry and biology, respiration is talking about taking glucose or energy molecules and then breaking them back down into water and carbon dioxide. So there are going to be different types of carbohydrates, and maybe you've heard these phrases before. Again, we're going back to our prefixes. So I think at this point, everyone's getting comfortable with it. We only really have three. The most basic type of carbohydrate that we use for food is going to be a monosaccharide, so one sugar. And it'll be the simplest of the carbohydrates. So I just have a sketched out molecule here. Don't worry about the structure right now. Just treat it as like a unit. I don't care what it looks like right now. We'll deal more with what carbohydrates look like in the subsequent slides. But when we see that there's only one molecule, if you treat it to try to break it down any further, it doesn't break down. And so that's what we're seeing here. We can either use some sort of acid or an enzyme. I'm making a generic reaction. It's just saying I got one thing and I can't break it down any further. That takes us to the idea of disaccharide, literally two sugars. And it only consists of two molecules of sugars that have been covalently bonded to each other. And that's what I've shown down here. So there's our disaccharide. Again, don't really worry about the connectivity. Just kind of think of it's like two things stuck together. And if I treat them in such a way that I could break them apart, they break apart back down into two monosaccharides. And then the last one, so it's funny, we only really, well, you can use more prefixes, but these are the more common ones. The last one we're going to stop on is polysaccharides. And these are going to be the ones that contain many monosaccharides. Poly doesn't necessarily indicate like, is it 5 or 6, 500? right? Poly usually means actually quite large. We're talking 400 to over 1,000 sugars connected together. We don't discuss it here. I'll just say the term. The in-betweener between like disaccharide and poly is, you might have heard the term oligosaccharide. Don't worry about it. It's just kind of the in-betweener. Oligosaccharides are kind of like in that 10 to 20 sugar range, and then polysaccharides are for like big amounts of sugar. And we'll talk a lot more about that later, but typically these polysaccharides, this is how humans, animals, organisms, plants store energy for the most part, other than in the form of fat. So let's just go a little bit through. There's going to be a lot on monosaccharides, a little less on di, and then kind of just like a brief overview of poly. So mostly the monosaccharides will deal, this will be like the harder chemistry stuff. And then we'll more kind of transition into more just like, here's a molecule to be familiar with, here's what disaccharides look like, here's the function of polysaccharides. So monosaccharides, it looks weird right now. You may have not seen sugar, if you've ever seen a picture of sugar before, drawn the way that these two molecules are shown here. But in general, what a sugar is going to contain, right, is either going to be an aldehyde or a ketone. So that's why we're introducing this chapter after our last chapter, right? And then what we have is a chain of carbons afterwards. And we're going to expand our definition a little bit more after that. And in fact, what it means when it's added to water, it will actually wrap around and form a circle. We'll get to that. Right now, we're dealing like, what if I had a sugar and it was dry on my desk, right? So typically what we see from monosaccharides, they don't get giant, but they're usually between 3 and 8 carbons long. So again, don't get confused with, monosaccharides, disaccharides, right? Those are the number of sugars connected together. Each individual sugar will typically have between 3 to 8 carbons. And like it says, has to have one of those carbonyl groups, like an aldehyde or ketose. And so to add more definitions, but it helps to clarify because these are commonly used words. Any sugar that has ends with an aldehyde will be called an aldose. Pretty easy. We can see that it's ose typically indicates that you're dealing with a sugar. So we have an aldehyde sugar. And so that's why you see something like erythrose is an aldose. If we have a ketone, like the molecule we see on the right, it's a ketose. And that's what we see here. We have actually the same number of carbons. So it's going to be erythrulose. So it kind of changes the name a little bit, but we're not worried. We're not going to deal with naming like that. So don't worry if that freaks you out. Right now, we're caring about aldose and ketose. We just see it's a sugar with a ketone. We'll call it a ketose. And the last thing, right? This is what really distinguishes, a like a sugar group, a carbohydrate, from just any old aldehyde. It's that at the very end of the molecule, you'll always have one of these hydroxyl functional groups. So that's going to be the key we're looking for. So if we want to kind of summarize what we're looking for, if we're trying to identify a molecule as a carbohydrate, number one, do we have an aldehyde or a ketone? Do I have between 3 and 8 carbons? And does that last carbon contain a hydroxyl group? And that's going to be our definition. So how do I identify any old aldehyde or ketone versus, is it a carbohydrate, a monosaccharide? So like I said, we're not going to go too deep in this. This is about the end of our naming. Other than that, I'm just going to, tell you, we use the common names. Don't want to like overwhelm because it's a new, again, it's funny, you get disparate branches of science that are finally coming together. They all have different naming conventions. So this is more of like a biology naming convention. This one is just we're gonna have to memorize. It only goes 3, 5, and 6. We'll stop at that. But if we have something with 3 carbons, it's a triose. Anything with 4 is a tetrose, pentose, and hexose. So again, it's nothing really too new. We just see that we have our Greek prefixes, and then we just attach "-ose" on it to mean that it is a carbohydrate or a sugar. So let me give you some examples of what that looks like, right? So these are some common carbohydrates, And again, we're seeing those, when what we're looking for in terms of identifying, is it a carbohydrate? Is it a sugar? We notice all of them have either an aldehyde or a ketone. They're 3 to 8 carbons long, and they end the molecule with an alcohol. So we're good. And so if we look and go through, so something like glyceraldehyde, right? 1, 2, 3. We have 3 carbons that make up that straight chain. So we know we're going to have a tri. We have an aldehyde group at the end. So we'll add in our aldo to indicate that it is an aldehyde. And now that we've met all the requirements for a carbohydrate, ose. So we can see how it breaks down from the structure to just a generic term, an aldotriose. It doesn't really tell us what the compound is. Like I said, we're not going to focus too much on naming in this particular chapter because it can get very complicated, but it's more identifying, I look at the structure of the molecule. Can I indicate, is it an aldotriose? Or is it something like an aldotetrose, which would be the next molecule? Why did the name change? Well, we can see there's an additional carbon, and that was really the only difference. Then I'll kind of skip and we can do ribose. Ribose is an important one. So when you think of DNA and RNA, oops, can't spell. Not DNA. Well, actually, yeah, we can do that too. But RNA particularly, that R stands for ribo, right? It's standing that the backbone of that molecule, and when we get to chapter 17, we'll deal with the structure, but DNA is made out of a long chain of sugars, and that sugar, for RNA in particular, is going to be ribose. For DNA, it's deoxyribose, which means it's missing one of the hydroxide groups. Off the top of my head, I don't know which one. All right. My point was, I only wanted to mention ribose because it'll come up a lot later, but fructose, the last thing I'll say on this slide, I know I'm beating it to death, right? We can go through the same exercise like I did with glyceraldehyde. We can see that we have a ketone, so we know it's going to be a ketose. How many carbons do I have? 1, 2, 3, 4, 5, and 6. So it's a ketohexose. So hopefully it doesn't get any harder than that. Not hopefully, that's as hard as it will get for types of problems I'll give you. Just hopefully it's not difficult in the long run to study. All right. So what we've done now is we've introduced the basic concept of what carbohydrates are. Before we can start to talk about more of their properties and and their structure, especially the structure when it will be biologically active, we need to start dealing a little bit more with some harder chemistry concepts, and chirality is going to be that. And it seems simple because it is, but sometimes when you actually have to do it, it can be kind of a challenge. So I'm going to try to be as clear as I can in the explanation, and then be very clear on the types of problems I would give you for something like this. So what chirality means, it actually translates from Greek to mean handed. So I think it's kopto or chiral, something like that, means literally hand. And so it's the handedness of a molecule. So that kind of sounds weird. So we will call them left-handed and right-handed. And we'll learn how to identify if a molecule is left-handed or right-handed. But what the major idea is going to be is that if you take something that is chiral, that has handedness, so I'm gonna have to turn on I have to be able to see myself, hopefully. Why can't I see myself? All right, I'm gonna have to do it this way. Oh, I didn't realize I was out of center, guys, sorry. There we go, I think. Why can't I do that? All right, so for handedness, right, you have, let me see if I can do this. This is incredibly difficult working backwards. All right, so we have handedness, right? I have a left hand and a right hand, and it just looked like I think it was right, and it's opposite, don't worry, right? But the idea is if I keep them in the same orientation, right, I cannot superimpose them. There's no way, because on this side I have a thumb that's wagging, I don't know if you can see that, and this side is another thumb, and those should be lined up. But instead I have a pinky on this side, I can't do that, independently move them, right? I have a pinky on this side and a thumb on this side. So despite both being hands and looking like they're very similar, there's no way I can overlap them. And that's why they have different functions, right? You can grab things with your right hand and your left hand, but your left hand doesn't curl up back into itself, right? On the other hand, if you took, I can't do this, I don't have a mirror, but if we look on the picture on the screen now, right, if you did take a right hand and you took the mirror image of your right hand, they would superimpose, right? And so we'll talk a little bit more about that. It gets weird. I thought it'd be easier to use the, to show it with my hands, but apparently working in reverse is not easy. All right, now it's not letting me switch. There we go. All right, so we're going to come back. I'm going to give you some more explicit examples on chirality. That's more just an introduction. So why would a molecule be chiral? Why would a molecule, you give the structure, so why does it make a difference if they can't, the mirror image doesn't stack on top of itself? So this is going to bring us into, we've touched on this briefly about isomers, right? We talked about, I forget which chapter now. I know we did talk about it, but again, Greek iso meaning same, and mer being, I'm just going to call it parts or things. Keep it simple. I don't know if that's the real translation. That's the way I always think about it. So it's a molecule made up of the same parts. So what do I mean by that? So if we took a generic, C2H6O, and I'm saying those are my only building blocks, what could I build out of it? So I could build something like ethanol because I have my 2 carbons. Then I could add in my 6 hydrogens. So there's 3, 4, 5, 6. And then I can use my 1 oxygen. So that would be one way I could use C2H6O to build a molecule. In that case, ethanol. And we can see, distinctly it's an alcohol. There's also another way I could arrange this. I can use 1 oxygen. I can use my 2 carbons, and then I can use, what color did I use for hydrogen, right? 3, 6. So in both cases, ethanol and dimethyl ether have the exact same chemical formula, but depending on how I connected the molecules in 3-dimensional space, they have very different reactivities, right? One of them is anesthetic. So dimethyl ether, it's kind of toxic, it will knock you out. It's also a gas. Ethanol is a liquid. It will also kill you in large amounts, but that's effectively what drinking alcohol is. So very different chemistries despite having the exact same parts. And so that leads us to the idea, and we've done this is why it's a brief review, of a structural isomer, right? And then you can go and you can prove to yourself later, but for the sake of time, I'll leave it here, but you can trust me on it, that if you took, a different chemical formula, so C3H6O, there'll be different ways that you can connect those molecules in 3D space. In one case, I can get an aldehyde. The other one, I can get a ketone. And we've already seen that ketones and aldehydes are going to react very differently. So that's the first kind of thing that we need to get under our belt before we move into talking about chirality. Oops, I guess I went backwards. Go forwards. There we go. So what does a chiral molecule look like, right? And so why, and we'll get to the end, I'll give you a very good example why it matters, because in biology, well, boy, I can even tell you now, in your body, you have one handed type of amino acid. Humans and almost all animals only use L amino acids. Bacteria occasionally will use something called D amino acids, which is actually a chiral mirror image of the amino acids. Your body is so sensitive because it doesn't use D amino acids. It's actually a signal to your immune system that there's a bacterial infection. And it's because a D amino acid behaves very differently than an L amino acid. So it's kind of like a heads up. For me, we're not, at least in this class, I'm not going to have you make large molecules. I'll keep them to something about this size, maybe a little bigger, but not much bigger. So we're not going to go to large chiral molecules because they get very confusing very fast. So let me give you an example, because I know I used a hand example. So I mean, you can see it, but sometimes it's easier, I think, to see with a chemical example. So what I have shown here is just some random molecule. This is the one almost all textbooks use. We just take a carbon, which is in the center, and then jam 4 different things attached to it, usually hydrogen plus 3 of the halogens. So we can see on that molecule, I got 1 hydrogen, a chlorine, an iodine, and a bromine attached. And it's also shown with correct three-dimensional orientation as well, right? So we can see it's making that tetrahedral shape. So it's not a flat object. If I took this molecule, right, the one on, sorry, again, over here, and then I put it in the mirror, just like I was actually having issues with my hands because I'm seeing my camera, which flips the image. So I'm getting it backwards. So technically I was seeing my own mirror image. So all I did was change the conductivity. So it's a mirror image. And what we see then, if I try to put this molecule back on top of it, like in three-dimensional space, just like the hands, what we see is, yeah, some of them overlap, like the hydrogen does, the carbon does. But look here, there's a clash between bromine and chlorine, right? That's indicating that it is a chiral molecule, that the mirror image is actually going to be chemically distinct. And they're actually finding out even more. And it's become more common now that we've got better techniques to separate molecules, that a lot of old medications used to be a mixture of two chiral compounds, even though they'd be chemically identical. And they're finding out that your body sometimes preferentially will, there'll be more of a therapeutic effect from one type of a chiral compound versus its mirror image. So it actually is very important in terms of reactivity. So we have a phrase that we use to indicate if two molecules, if the mirror images don't superimpose, and that's going to be enantiomer. It's just a word you're going to have to know. I've been trying to hold off saying it until I introduced it, but that would be like here, right? These two molecules here would be considered enantiomers. Why? Because their mirror images cannot be superimposed. So I guess that's probably the take-home message. So if I was going to give you a question like this, say on the exam or something like that, I would give you a simple molecule and typically ask you, is it chiral or is it not? And then, it's going to, sometimes you have to just do it in your head, because usually what I would do if we were in class, I have model kits that I can give you, but we don't have model kits that I can like mail you or anything like that. Sometimes it's helpful to use your fingers. I'll make the kind of a tetrahedral type shape. Let me see if you guys can see. Let me get out of this. I want to make sure I can see you guys again. Right? I don't know why it won't. It's been really, the camera's at a weird angle today because like I'm actually sitting at now about a 20-degree angle to my computer. And it looks like to me it's straight. I can't tell. Let me see if I can make that better. Oh wait, no, I was just cut off. That's why. All right. So ways that I've done it when I've had got stuck in the middle of like lab or something like that and trying to figure things out, it is possible to try and use different colored pencils, though it's not easy to hold. But you can try to think of ways that possibly could help you figure out, if I made an mirror image, would they be superimposable? And I'm not going to make it a major focus, but I'll guarantee you there'll be one type of problem on that. But I'll make several, example problems so you know exactly how it would be phrased, exactly what I'm looking for, and then gauge that level of difficulty. All right, so I've been doing a lot of talking because we do need to get through a good amount of material. Is there anything so far that's throwing people off, need a reclarification?
I have a quick question.
Yeah.
So with the chiral molecules, which one would you call a chiral, or would you call both of them?
So it's we're learning definition. That's a really good question. So they're both chiral, and they would be enantiomers of each other. So
Okay.
that's how the way you kind of phrase it.
Okay, cool. Thank you.
And it doesn't even have to be a molecule. I removed a slide I made because I was trying to condense, I'm trying to make this so we can get through the chapter as quickly, not quickly, but as efficiently as possible. There are, you can ask yourself in real life, molecules, I mean, when you have gloves are chiral. They're not molecules. Baseball bats are achiral because every baseball bat doesn't matter. You can see it in the mirror. It can still be superimposed, right? It'd be like taking a pen is not chiral because if you look at the mirror image, I could immediately superimpose a pen over it, but like gloves can't. So chirality isn't just for molecules, though in most cases, that's how almost everyone uses it. All right, it's keep being, there we go. So that gives us to the idea of an achiral molecule, right? Again, we can do that same exercise, and I don't know if we've actually run into this yet. I might use it a lot. When you see the Greek prefix a, it means not. So it's the opposite. So it means not chiral when you have achiral. That's why we hear a lot of people talking on the news about being asymptomatic. That means not symptomatic, not showing symptoms. And so you can see here, right, we have our starting molecule. You look at the mirror image of the starting molecule. And then, but if we try to superimpose, yeah, they don't feel like they fit, but I could rotate it in space and eventually get back to the same molecule. So that's one thing I didn't show in the last slide. It's difficult to show without animation, but you can try to prove to yourself too. There's no way we can rotate this molecule in space. It will never, ever line up. That's what's making it chiral. The achiral one, I could do some kind of transformation to it, which means move it in space. Man, it's being delayed this morning, right? And then they would superimpose. So this is where I was kind of doing that foreshadowing of why biological systems are typically designed to only operate with one type of enantiomer. And we'll start with carbohydrates, but we'll find out amino acids also have chirality. DNA has chirality as well. And so your body only typically deals with one enantiomer. We actually physically don't know why nature would evolve like that. But physicists are trying to figure that one out, but we may never know. All right. So basically what this is kind of getting at is, maybe in biology in high school, you were introduced to the idea of lock and key mechanisms on enzymes or proteins. So basically it's like a big structure and it has little, places in it that another molecule could come in and bind with. And that's what it's showing here. So this carbon here is chiral. And we can see in one case, if I kind of plug that into the enzyme, it's going to click in. And so the body would recognize it. If you take the mirror image of this molecule and then try to click that back into the same enzyme, it's not going to work. And so that's kind of the biological mechanism of how a chiral molecule can be detected by your body. Like I said, we're going to do more of that when we get to into proteins, which will be, I think, in about a week and a half-ish or a week. One last thing I'll say that it's kind of not a cheater thing. This is what most people use to help find out if something is chiral or achiral. Something is automatically chiral if that center carbon's connected to 4 different things. And I'm going to put that in quotes. What those things are, it doesn't matter. As long as all of them are different, then it's going to be chiral. If you have, let me actually go back to the last slide, right? We notice here, right, and that kind of, it fits with our pattern. Come on. Right, we see that that center carbon, 2 of the same things are the same. That's an indication that something might be achiral. Or actually, that is the definition of achiral. It is having 2 of the same things connected to a carbon. So that's a good way also to determine if something's chiral or not. So I've kind of like tried to divide this into different parts of the lecture. So that first part that I was talking about is mostly, introducing what carbohydrates are, introducing chirality. What I want to do now is join those 2 ideas together and start talking about how carbohydrates are chiral molecules, and then how we go about naming them, and then some of their functions in the body. So for the most part, your body recognizes disaccharides. We'll get into a second what D means. It literally stands for dextro, which means right-handed. But like I said, we're going to get into that. So let's get into that. So one way that we're going to look at is how, and we kind of already did this a little bit, how do we represent sugars in space? And how do we determine if, because they can get quite long, 8 carbons long, how do I determine if a molecule is going to be, chiral or not? And I'm going to tell you and foreshadow, it goes into 2 categories, L or D, which is kind of dumb because L stands for left, D stands for dextro. I don't know why it's not sinister and dextro to keep the Latin the same, but that's what we use. So the way I always tell my students to remember it, pick one, L. L is always left. That means D has to be right. So I'm not going to have you draw Fisher projections. Usually we would do this in class and it'd be like an exercise we'd do together. But now that we're separate, all I really want the take-home message is going to be is to recognize Fisher projections, and then I'm going to make I'll indicate exactly what I'm looking for in terms of understanding. Right. So what a Fisher projection is basically taking something that usually has implied three-dimensional shape. Like those wedges are telling me those wedges here, right? They're telling me that that hydroxide and the hydrogen are coming at me, and those dotted lines are saying things are going away from me. And we've seen that when drawing 3D structures. So what a Fisher projection is basically you take that molecule and just theoretically just crush it onto the page and then see what happens, right? So what this helps us find out, one, it can help with that naming. Is it a triose or an aldose or something like that? So we can easily see we have our carbonyl. We can easily see we have that hydroxyl. So we know we're dealing with a carbohydrate, right, a sugar. What it really helps us see, though, is if a carbon is chiral or not, right? So I have that carbon in the center. And so really similarly to how we represent the skeletal structure of molecules when we draw them, we don't explicitly label where the carbon is. The same thing happens with the Fisher projection. Any of these intervening carbons in the middle, like the one I have highlighted in yellow over here, right? Those ones don't get a C representing carbon, only the terminal ones. But my main point, what I was saying, you can tell if something is chiral or not, right? The easy way with the Fisher projection, we go back to that rule that we're looking at a carbon and if it has 4 different things attached, then we're going to say it's chiral. So if I take a look at this molecule on the left-hand side, right, let me just erase everything. Right, I can see that I have an aldehyde, that's one thing. I have a hydrogen connected to that carbon, that's a second thing. I got a hydroxyl group or an alcohol group connected, that's 3 things. And then I have whatever this group is down here, and that will be 4 things. So I know that carbon in the center here, the one again I'm highlighting, is going to be chiral. And you can do the same exercise over here as well. So there's our aldehyde and H and OH and then that group. So both of them are chiral. So they're structural isomers. You could go through and prove it to yourself because they have the exact number of carbons and hydrogens. And they're stereoisomers because they're all arranged in the same way, but there's a little bit difference in terms of their handedness. So the other thing that's important to pull out from the Fisher projection is if something is an L or a D isomer, and it actually is pretty easy. The only thing you have to look at is the last carbon, not the very last one, not this last group down here, maybe I don't know how to phrase it, but the last in that middle chain of carbon, so like I'm showing here with erythros, right? That's the one we really care about if that carbon is chiral or not. It actually dictates if the rest of the molecule is going to be a left-handed molecule or a right-handed one. And it gets pretty easy. So you recognize it's chiral because I have, again, I don't care how big the thing is. I'm going to count that as 1 thing. Then I have my hydroxyl group, that's 2. Hydrogen's 3. And then the last bottom group is 4. And how do I determine the handedness? You look at the hydroxyl group. Hydroxyl is on the left. That's why I have my L isomer. Literally stands for left. If we look at the other stereoisomer of erythros, right, we can do the same exercise. You're looking at that last carbon down here. I'll put it again, I'll highlight it so we know we're on the same page, right, that carbon. So I got 1 thing, that big giant group on top. Don't care how big it is. I can just count it as 1 thing because it's going through 1 bond, right? There's a second thing, a third thing, and then my 4th thing. So we have our chiral carbon. It's on the right-hand side of the molecule, that hydroxyl. And we actually use the Latin dextro. that literally means the word right. So when you hear the word ambidextrous, right, it means it's like using your right, because this is the old word, right? It doesn't matter if people are right-handed or left-handed, but that's where ambidextrous comes from. It means you can use both hands like you're supposed to, the right hand, quote unquote, right? It's actually where the word sinister comes from. Sinister used to not actually mean evil. It just literally translates to left-handed. Apparently people really, really didn't like lefties back in the day, but those words have stuck around. All right. So take-home message. If I gave you some sort of Fisher projection, could you tell me if it's left-handed or right-handed? Could you tell me if it's D or L? That's the take-home message. You look at that last carbon down here, you look at the hydroxyl group, does it point left, does it point right? That's what I want you guys to come away with. So any questions on determining D versus L stereoisomerism? All right. So again, these are just a bunch of examples, but we still got 10 more minutes, so we're doing pretty good. If we don't get to the last part, I'll make a small video on it. It's more just, for the more you know type slides, talking about polysaccharides. I will type something up telling you exactly what I want you to read on that, but it's mostly just like recognizing what glycogen is or something like that. But that's if we don't get to it. So things that I want you to become familiar with, though, and this is because I picked out certain molecules that you will see as a nurse, right? So I do want you to memorize, I'm not saying you have to memorize their structure, but I want you to memorize kind of like what their functions are, and like basically these 3 bullet points for each of them. And there'll be 3, I ran out of space, so galactose is on the next slide. But D-glucose, right? So this is what when we say like simple sugars, this is what we're talking about. So when you're saying, oh, I need to get sugar quick, I'm hypoglycemic, or if, you haven't eaten all day, you're saying, oh, eat some simple sugar. That's what we're talking about. Eat glucose, fructose, corn syrup, and honey are all made out of glucose, right? So another name, and I want you to know that too, is going to be dextrose. So that's a common name. I don't know why, but I think it goes back to the 1800s when they didn't know if there was a difference. I don't know. I'm kind of talking out of my butt on that one. But glucose has 2 common names, D-glucose and dextrose. It's also called blood sugar if you're dealing with, if you're taking like blood or something like that. So when you talk about your blood sugar levels, they're talking about your glucose levels. The reason for that is glucose is the main energy source for the body. So that's why if it's already in the form of glucose, your body doesn't have to modify it at all. It can immediately use it for energy. Oops, D-fructose. This one you don't really need to know. I don't know if you need to know this one as much for nursing, but it is a common sugar. It's twice as sweet as glucose. And so that's when you hear like high fructose corn syrup. What that really means is you've taken a molecule and you've treated it with an enzyme, and it's tried to approximate what honey looks like. It's funny because when I was younger, I never used to believe in that hype. And I'm like, your body shouldn't care the difference between honey and high fructose corn syrup. It's actually been shown with evidence that, yeah, high fructose corn syrup does behave differently in the body. But I know there's still, I'll put it that way, it's still anecdotal, but there are papers out there starting to come out that high fructose corn syrup does behave differently than honey. And the last one I want you to memorize is going to be galactose. Just because it's a common, again, it's going to be a common sugar monosaccharide that comes up a lot. We don't really see it free in nature. It's usually bound up, and this is when we get to disaccharides, as lactose. So that's the sugar that's in milk. And then some people do actually lack the enzyme to break down galactose, because usually can convert lactose. Certain people are lactose intolerant, some people aren't. But usually you can break down lactose into glucose and into galactose, and then your body takes the galactose and then converts it to more glucose. So that's why some people can eat lactose and be fine, because they have lactase and they have another enzyme that converts the the galactose back to glucose. So long story short, right, I'm just more looking, recognize the names. You don't need to know the structures, but some of those, bullet points might show up as like a multiple choice or something like that. So this might be, well, we'll see where we get. We still got some time. The last thing I want to do is show you another way to draw the structure of sugar. And this is the more common way you'll see it. So if you Wikipedia this or do a Google search for the image, what you'll actually see is something that looks more like this molecule here. And the reason for that is, even though we've been using those Fisher projections that show kind of a straight line molecule, when sugar is dry, it looks like the straight line molecule. When it gets dissolved in water, it actually, what's going to do is, it's called a reaction, it's a cyclization. So it literally makes a ring. I'm not going to have you convert between the two structures. I just want to show you so you can kind of visually see what's happening, that they're representing the same molecule. So we'll take glucose. And so what I have on the left-hand side, right, this is our Fisher projection of D-glucose. D, because that last hydroxyl on that last chiral carbon, right, is pointing to the right, so we know it's D-glucose. What actually ends up happening, and so just do in your head, I mean, I'm doing it on the screen, all I did was take the molecule on its basically standing up, and I laid it down. When you add this to water, remember, molecules aren't rigid. They're actually constantly moving with random motion, and if you get long enough, they can twist and bend in space, kind of just like, like headphones or something like that when they get long enough. What ends up happening is actually you can get a chemical reaction on the same molecules reacting with itself. The alcohol group, that last chiral carbon that's here, that oxygen that determines if it's L or D, right? It's actually going to, and we use the term attack, that's actually the scientifically correct way. It's attacking the aldehyde group. What ends up happening is that oxygen forms a new bond with carbon, and we're not going to get into it, but this gets rearranged into a new alcohol group or a new hydroxyl group. So what I would do is, if it didn't make, if you had a hard time following that, maybe come back to these slides or maybe re-watch what was going on. But it's basically a molecule's wrapping around itself, attacking itself, reorienting itself, and then it's forming a new bond. And so that actually happens once it's added to water. The thing is, though, if we go back actually and look at this structure, and I gotta see so I can see myself again, so I can use my hands as a tool, right? So I'll try to do it the same way so it looks almost the same. Whatever. All right. Use your imagination then, because imagine this is like an aldehyde group, the one that is here, right? Because of this single bond, and let me change the color on that. Because of that single bond here, that actually can it paused. I can't see myself, but I have to undo that. Right, it can actually rotate in space. So sometimes that H can be pointing up, sometimes the H can be pointing down, right? So it actually, due to random motion, it'll be constantly flipping. Because of that, and it's going to be a little bit random when it does happen, but let's see. Come on. Right? That means when that reaction happens, sometimes it can lock the hydroxyl being down once the reaction has happened, and other times it can lock the hydroxyl being up. And so we need to indicate if that last, so if you haven't noticed too, in the book I think clarified, alcohol and hydroxyl mean the same thing. So just OH. When the hydroxyl group gets locked in the down position, right? So that's what I'm seeing here. We're going to call that the alpha isomer. And this one always throws me off because when I was learning this myself, I still remember thinking like alpha, I want it to be top, but it's not. Alpha is down. So we had our glucose. We knew it was a D isomer because we saw that in the Fisher projection. And then from the Hayworth projection, we can find out it's an alpha isomer. And we can do the same thing over here. There's my glucose. It was D-glucose. I got that from the Fisher projection. Now my hydroxyl group is now in the up position. So it is the beta isomer. So beta D-glucose. And so I don't know if you've heard phrases like that before, alpha D-glucose, beta D-glucose. right? That only happens once it's added to water. You can't get alpha or beta D glucose like on the shelf. It's just going to be, a flat molecule. There won't be alpha or beta. So like I'm saying, take home message. I'm not going to have you convert between Fisher and Hayworth. What I want you to be able to do though on a Hayworth projection is figure out if it's alpha or if it's beta, and then recognize that it is a cyclization reaction, that the molecule is reacting with itself. Those are the two things that I wanted to take home. And so basically this is just a summary slide of that. So what actually happens once that reaction, it goes to alpha or to beta, it's not over actually. What ends up happening is it's in equilibrium. So as soon as that alpha isomer gets locked down, occasionally it just splits back open. It'll turn back into regular D-glucose. Maybe that aldehyde group flips around a little bit. And then just by coincidence, it reacts and turns into the beta isomer. So it's constantly switching back and forth. So you can't just you can never ever get pure alpha D-glucose or pure beta D-glucose. That's impossible. The reason for that is because that reaction is constantly happening and undoing and then flipping around. We're not going to get into this too much, but it's not a 50-50 distribution. The reason for that is it just so happens that it's more favorable to form the beta isomer rather than the alpha isomer. That's something that gets a little bit deeper. We might talk a little bit more about that when we talk about proteins, but it has to deal with something called steric hindrance. You don't need to know what that is. It just means sometimes it's easier to react if the molecule is in a certain orientation, where it's not big and bulky and stopping things from reacting. An easy way to phrase that is just saying it's just easier for the reaction to happen to go towards beta D-glucose than it is to go towards alpha. And that's exactly what I'm showing. Oh, the last thing I wanted to show here. I think this actually ends this part 2 thing. So give me my last minute and then I'll let you guys go. I just wanted to show you that fructose is also, you could do this with some sort of ketose, and it doesn't have to be an aldose that does the cyclization reaction. A similar thing happens. We have that last chiral carbon that has the hydroxyl group. Instead of attacking the aldehyde, it can now attack that carbonyl carbon in the ketone up here, and this similar process happens where it cyclizes. The only difference is now you're 1 carbon short because they're both hexoses, but one of the carbons is bound up at the top. So it actually makes a pentagon rather than a hexagon in terms of its shape. I just wanted you to see that and recognize it. Beta and alpha also still apply. Again, the only thing is it makes it a little bit more challenging. Got to remind yourself, you're looking at the hydroxyl group, right? Hydroxyl down, alpha. Hydroxyl up, beta. All right, great. So that's actually a good place to stop. I'll make a short video and probably type something up because it might just be easier to give you a document on it. It's just talking about polysaccharides, what their functions are in nature and in the human body, and that's pretty much about it. All right, everyone. So we're good. I'm going to stop the recording now. So let me just make sure I don't mess this up. Let me get out of the screen share mode first because I don't want to mess things up. What? So they updated all the software now, so everything's moved. All right, I'm going to stop.